Hello students, now we will learn about seed and its structure. Seed can be defined as matured, fertilized ovule. When ovule is fertilized and it developed in further, then it will get converted into seed. Study of seeds is called spermology. Seed has cotyledons inside and outermost seed coat. Seed coat existence is common in both dicots and monocots, but the other layers means how many layers of seed coats are present that vary from dicots to monocots. Seed has outer seed coat commonly in dicots and monocots, but number of layers vary between dicots and monocots. In case of dicots, seed coat has two layers. That is outer testa and inner tegmen. In case of monocots, the seed coat is fused with pericarp. In monocots, seed coat is fused with the pericarp. Now, in case of dicots, what are the structures of ovule that are giving rise to testa and tegmen? In ovule, we have one special layer called as integuments. Those integuments have been modified into testa and tegmen. These are the developed structures, developed layers of integuments of ovule. In case of developing seed, what are the structures? Seed has cotyledons, like dicot seed. This is embryonal axis. This part is embryonal axis. Embryonal axis has, in case of dicot seed, epicotyle and hypocotyle. This part is epicotyle. And here at the bottom part, this is hypocotyle. This tip part is plumule. This bottom part is radicle. Epicotyle helps plumule or the shoot part of the developing seed to break soil and move out during germination. Second one, hypocotyle pushes radicle towards earth layers. These are cotyledons. Cotyledons are stored food material. Stored reserve food of a seed. Now, development of this plumule will be towards the chalaza of the ovule, and hypocotyle will be towards the micropyle end of the ovule. Coming to cotyledons, we'll write one more point. Cotyledons in dicots are 2. Next, cotyledons in monocots is 1 papery like sheath called scutellum. We will see which parts of ovule have been converted into other parts of seed. So we will write 2 integuments, embryo sac, this is ovary, this is ovule, here will be placenta, and this is funicle. This junction which is attaching funicle to the ovule, this is called hilum. Above the hilum, up to the point where integuments are touching, this is called raphe. And this opening is called micropyle end. This is typical anatropous ovule. These are integuments. This is embryo sac. This is central cell of the embryo sac. These are polar nuclei. These both are polar nuclei. Now, endosperm, which is present here, is also the reserve food material even after fertilization. So we will write reserve food. Endosperm. Before fertilization, the reserve food for the ovule will be nucellus. So here we have seen the terms like integuments. Those integuments will be the outermost seed coat layers, which are converted into testa and tegmen. Here 2 terms we have written, epicotyle and hypocotyle, and also other term we have given embryonal axis. These both combinedly are called tigellum. Epicotyle and hypocotyle combinedly are called tigellum. So these structures, hilum, funicle, micropyle, what happens to them when they are converted into a seed-like structure? In some seeds, we see some whitish color-like structure here. That is called hilum. And raphe's position is here. Here will be micropyle. So the outermost will be the seed coat, innermost will be the cotyledons, which are reserve food material in case of seeds.